Hi there. Can I just check that you can hear me okay? Can you just let me know in the chat, please, that you can hear me? Perfect. Brilliant. Okay, so hi, my name is Thea. I'm going to be teaching you the biology content on this course. Um, I didn't want to start before 6 because I know if people are joining at 6, they feel a bit rushed and pressured if the meeting's already started. So I try and start on time so that no one feels stressed. So I'll talk to you a little bit about how we're going to approach these sessions. What I will do is I will cover the sessions in a lecture-like style because you've got a tutor to go through the work with you in a tutorial like style. So I'll teach you in lectures because that's what you're going to get when you get to university. So it's important that you get used to that method of teaching. It does feel quite fast paced sometimes when you're not used to it, but I have provided these lecture slide packs for you. So you don't need to be furiously scribbling down. You can just pay attention and make sure you can understand it and go through the content at your own pace if you want to go back through it again afterwards. One of the important study skills with lectures is to be able to listen to them rather than try and write everything down so that you can work out what the key points are. So what I'm hoping to achieve in these sessions with you is to go over the information with you, hopefully iron out any misconceptions and to focus more, I suppose, intently on the content that is going to be in your assessments, because obviously I assume you've had a little look already at the course content on and had a little bit of a read through or a flick through of what you've got to cover, and you've realized there's an awful lot of material there. There's a lot of reading to do for you. So hopefully these sessions will focus on the stuff that's important for assessment and also iron out any misconceptions with some of the hard topics, because some of it is quite challenging. And don't worry if you do find it challenging, because it is hard. It is a big jump up to this level. Okay, so we're going to go make a start. So this is session 1, and the unit what we're looking at first is cells, tissues, and organs for health. See. Sec. My slideshow buttons aren't working. I've got to do it that way. So that's the unit number for you. And the learning outcome, or the point that we're looking at today, really is to understand the structure and function of a generalized human somatic cell. So somatic, that's probably a new word to you. It's not an everyday word that we use in general conversation. And we'll have a look at what that means in a minute. Okay, so session 1, we'll probably take 3 sessions to cover this content of the course, this unit. So one of the things you need to be able to do is to discuss the structure and function of key cellular microstructures. So that means the things that make up the cell, the microstructures. You might have heard them before if you remember back to school. If you've studied biology at all before, you might have heard them called organelles, maybe, because some of them are organelles. You might have heard of the nucleus, and you might have heard of the mitochondria probably when you were in school. You might remember the chloroplast from plant biology as well. That was another big one at GCSE level. So you've probably heard of some of these before. We're not looking at plant cells, obviously, because we're focusing on human biology, and humans are animals. And what we've got on the screen is a generalized animal cell. As we're going to see later, most of them are very specialized, so they don't tend to look like this at all when you find them under the microscope. But this is quite a general average cell that we're going to have a look at to start with. And animals are eukaryotic organisms. You can see that word at the top left of the screen there. The eukaryotic organisms, that means they're a more complex type of organism. They've got more complex cells. There's another type called prokaryotes, and we're going to have a look at them in a later unit. Includes things like bacteria. They're much more simple. They don't have these organelles, these complex structures inside their cells. like we do. So humans are eukaryotes, animals are eukaryotes, plants are eukaryotes, fungi are eukaryotes. They're eukaryotic organisms, and we're going to have a look at animal cells in particular. So there's two types of cells broadly that we can divide our cells into in animals. There are gametes, and that includes eggs and sperm. So these are quite special cells because they're the ones that are involved in reproduction and nothing else. And then everything else, completely everything, absolutely everything, your blood cells, your nerve cells, your epithelial cells, which we'll be meeting later, they all go into the somatic cells category. It just means everything else. It means an adult cell, shall we say. It doesn't mean cells that you only find in grown-ups, but it's been developed and has got a specialist function. So those are our cell types. Okay, so we're going to start looking at these structures, some of which are organelles, some of which aren't. And what we've got here is the first important structure that we need to be aware of. It's the cell membrane. So this is all around the outside of the cell. There's no cell wall. You might have heard of cell walls at GCSE. If you studied GCSE, you might have remembered there's a cell wall on some cells. Not on animal cells, there's not. So you don't have to worry about that. We've got just this membrane on the outside surface. And you can see on the right-hand side of the screen there what it looks like. It's very important because it controls what enters and exits the cell. It's made out of structures called phospholipids. You can see them in red and orange on the diagram. So this here is the phospholipid. It's a double layer. So you can see there's two layers of phospholipids there. Lipid is the term that we use to describe fats and oils. So that should tell you something. The outside of our cells is oily. And embedded in this sort of oily outside of our cells, we've got proteins. These are these blue grayish structures here. Now, some of them go right the way across the membrane. And that's really important because these have often got channels inside them. You can see one here. It's a protein channel. So that's one way that we can get things across our cell membranes. They can go through these protein channels, which are really, really important because there's some stuff we might want inside our cells. Water, for example, we might want inside our cells. We might want things like glucose getting inside our cells. There's also stuff we might want to get out. So there are various ways of kicking stuff out of the cells as well. So important structure, the cell membrane. It's on the outer surface. It controls what enters and exits the cell. Generally, if you're going to describe it, you'd say it's a phospholipid bilayer. So that's got two layers of these lipid fat-like materials and proteins embedded in it, which helps us get things across that membrane. Now, what this shows, right, I'm going to press play and really hope that it works. What this shows is a lava lamp video. So hopefully it works and doesn't take us to loads of adverts. No, it does take us to loads of adverts. Won't be long, 2 seconds, guys. Right, this is the important bit. I'm just going to put the sound down on the video. Apologies for the adverts. Right, this is important. I want to show it to you because look at how these blobs are breaking off from the lava lamp and separating really easily. But then look how they merge back in and reform back into one whole structure. This is exactly how the cell membrane behaves in our cells. So it's very easy for bits to pinch off that cell membrane, become a separate blob, which we'll call a vesicle or a vesicle. It can move around independently and then rejoin seamlessly with the cell membrane, exactly like this lava lamp is behaving. This is exactly the sort of material that we'll see, behavior that we'll see. Right, let's see if I can get out of that now. Not that easily, right. So what you can see here on this slide is a bit of the cell membrane. This is an actual cell, a real life cell, budding off, breaking off, just like we saw in that lava lamp video. It can pinch off and move somewhere else. This is actually a cell that's infected with HIV virus, and these bits are going to go off and infect another cell. But that's really important because it's one way things can get taken out of the cell, but it's a way we can bring things into the cell as well. We can merge in with bits. So lots of these structures within the cell, as we're going to see, are made out of this very same material as the cell membrane. So we've got structures surrounding the nucleus here. This is the nucleus. And we've got structures over here that we're going to have a look at. And they all contain this same membrane-like material and can basically interact with each other and reform. So we're going to start with the nucleus before we start talking about how these membranes behave. This is important. It is what we call the site of DNA. It's where the DNA is housed. We say it houses the DNA. And if you look at the screen there, it says it's continuous with the ER. That means it's joined onto. This is the ER here. This bit with these little dots on and this black bit here. ER stands for endoplasmic reticulum, and that's another long new word. There's loads of vocabulary in biology, and it's often worth going through and just having a vocabulary sheet after each session that you do, sitting down and reading, or after each lecture, and treat it a little bit like you would have your spelling test that you used to do in primary school. But instead of learning the spelling, learn the meaning of those words, and it will make it so much easier if you know what those key terms are. So endoplasmic reticulum is probably a new term for you today, as well as somatic cell. So the nucleus, it's where the DNA is found. It houses the genetic material, or the DNA, is continuous with or joined on to this structure here, the endoplasmic reticulum, that we're going to learn about in a minute. So it's got its own membrane, that same membrane material that we were looking at, like the lava lamps. And then this endoplasmic reticulum also is similar in structure. So this is an actual nucleus here on a cell. And if I just go back a little bit, you can see the nuclear membrane here. So the surrounding of the nucleus has got little holes in. And that's really important because this molecule, messenger RNA, can get in and out. So we can get messages from the DNA out to these structures around here. And those messages are instructions for building proteins. And we'll have a look at that in a much later unit. So there's our actual slide, micrograph. That means a picture from a microscope of a cell with the nucleus showing there. So the nucleus, as you can see on the top right hand corner, that's not a cell now, that's just the nucleus in this bit here. That's just the nucleus. It contains something called the nucleolus, and that's a spherical structure, just a sort of ball-like structure, which produces and assembles something called ribosomes. And we'll talk about them as well later on. They're really important in making proteins. And inside the nucleus, we find our DNA, and the structure that our DNA is found in is called a chromosome. So if it wasn't in a chromosome, it would be, imagine you took a ball of string, you unraveled it all and just chucked it on the floor on your desk. It would just be really tangly, really knotty, really big mess. And that's what would happen to our chromosome if it wasn't nicely, tidily wound up. So it's nice and tidily wound up in a chromosome structure. And you can see on the screen here, this is our double helix, the DNA, which you're probably familiar with. And that double helix is wrapped around these histones here. These are proteins. And then it coils up like an old-fashioned telephone wire. And then it's neatly packed into this chromosome structure. And that's to stop it getting all tangled and messy. So the DNA itself, if we were able to, we could see very small sections of it make up genes. And all that means it's a bit of code, a gene, that tells us how to make a certain protein. So our proteins are found, or instructions to make proteins are found in these sections of DNA, which are nicely wound around these structures called histones and wrapped up into this nice, neat structure called a chromosome. And then we find those inside the nucleolus, which is a spherical structure inside the nucleus of the cell. So here are some human chromosomes. This is what they actually look like. You can see they're not actually in an X shape here. They're just in a long, thin strand. They're only in an X shape just before cell division. So most of the time, they don't look like an X. They look like a line. And these are the chromosomes inside an adult human male, because it's XY. That's what they actually look like in real life. So what we've got here are the structures that I was just talking to you about a moment ago, the ribosomes. And these are tiny structures where proteins are made. Now, you can see my label is pointing to these bobbly bits on this structure that I mentioned earlier that I said was continuous with the nucleus. This is the endoplasmic reticulum, and it's got ribosomes stuck to the surface or bound to the surface. But we also find ribosomes free in the cell. We call this bit of the cell the cytoplasm. You might remember that word from GCSE possibly. It just means the sort of gel-like material that makes up most of the cell. It's a bit watery. It's got some stuff dissolved in it, like sugars and salts. And it's where you find all these structures. So free in the cytoplasm, we have these ribosomes, but also bound up to this endoplasmic reticulum, we have ribosomes. Not all over the endoplasmic reticulum. Some of it is not bobbly. You can see over here. This is called smooth endoplasmic reticulum, and this is called rough endoplasmic reticulum because it looks rough or smooth under the microscope. The ribosomes themselves are made out of a structure called RNA and proteins. And this is what they actually look like, just blobs really of protein. So this blue bit here, blue-gray bit here is one part of the ribosome, and this red bit is the other part of the ribosome. So we say it's got two subunits. This one's called the small subunit because it's small, and this one's called the large subunit because it's large. So nothing too complicated there. It's just made out of two chunks really that we call subunits, the large and the small chunk. And here we've got a micrograph image to show you what they really look like. And that there, RER, stands for rough endoplasmic reticulum. And the rough endoplasmic reticulum is this one here that looks rough because it's got ribosomes on its surface. It's quite hard to tell that it's rough or smooth unless you're used to looking at these images. They are quite tricky to get used to, but it looks a bit grainy, almost looks a bit fuzzy when you look at it. And that's how you can tell it's the rough endoplasmic reticulum. And that's the nucleus there next to it. So again, next to the nucleus, continuous with the nuclear membrane, joined on to the endoplasmic reticulum. So Here's our actual definition of the endoplasmic reticulum. They are sheets and folds of internal membrane, which may have ribosomes attached, and those ribosomes have a role in protein synthesis. So synthesis means to make the protein. They're also involved in lipid metabolism, so making lipids. And as we said earlier, we've got the smooth endoplasmic reticulum, no ribosomes, and we've got the rough endoplasmic reticulum, which has ribosomes. So far, we have got the cell membrane that's made out of this membrane-like material, which controls what enters and exits the cell. And we've said it's really important because it can form vesicles, if you prefer, that can pinch off like a lava lamp. And then we've got these structures, the nuclear membrane, the endoplasmic reticulum, which are similar in structure. They can pinch off and merge with various membranes within the cell. The nucleus, we said, houses the DNA, or it's where we find the DNA in these structures called chromosomes. And these structures here called ribosomes, many of which are stuck to the endoplasmic reticulum, have a really important role in making proteins. And the nucleus that contains the DNA, what that DNA actually codes for is building proteins. So here's the, if you like, the library with the plans on to build a protein. And these are the construction sites where the proteins are put together. This is a bit of a, I suppose, better image to visualize what it looks like. So here's the smooth endoplasmic reticulum. These are all membranes, no ribosomes, and this is the rough endoplasmic reticulum, which has got these ribosomes, these protein-making factories attached. And don't forget, this is surrounding the nucleus, which contains the instructions for making proteins, the DNA. So the smooth endoplasmic reticulum, the bit with no ribosomes, actually involved in is the bit that's involved in lipid synthesis. It's also involved in calcium storage and detoxification. And then the rough endoplasmic reticulum, it's got the protein synthesis ribosomes attached. So endoplasmic reticulum, sheets and folds of internal membranes, which may or may not have ribosomes attached. They're involved in making proteins and lipid. Next thing I'm going to have a look at is the mitochondrion. That's the plural, singular, sorry, mitochondria is plural. So if you've got two or more, these are really important organelles, which you might have heard referred to as the powerhouse of the cell. That's because there's a really important process that goes there called respiration. Respiration is how we get our energy from the glucose that we have in our food. So they're very important for energy, really, energy processing. They've got a double membrane. So again, we're talking about membranes, and that inner membrane is really, really highly folded, and that increases the surface area. And there's proteins attached to that, and they've got a very important role, which we learn about in a later unit. So I won't go into too much detail about them now. But they're quite significant in that they have their own DNA. So they actually tell us a bit of information about our evolution as well. That's not important in this unit. This is what they look like. You can see these mitochondria in the cell here. So we tend to say they're sort of, some people say they're sausage shape, but to me that implies long and thin. They're just not, they're a bit more rugby ball shaped, I suppose, than sausage shaped. But obviously if you took a cross section, so a rugby ball would be in that sort of shape, but if you turned it round and took a cross section, chopped it in half the other way, it's just going to look like a circle. So on a microscope, they might look circular, or they might look a bit sort of like a squished circle. So they're really small organelles, and it's where the cell gets its energy from for breaking down glucose. That's an image of the mitochondria. You can see double membrane, so you can see the outer membrane there and the inner membrane there. It's highly folded, we say. So it's what that does is it really increases the surface area. You can imagine plucking that inner bit out and stretching it out. It would have a much larger surface than the outer membrane if you took it out and stretched it. So by having this highly folded inner membrane, you're massively increasing the surface area. So that means you can fit loads more proteins and stuff on that membrane. So there's a lot more space because we folded it and packed it in. So that's really important to have that big surface area. But we'll learn about that in another unit. So another important structure is cilia. We're not going to go into that in too much detail here. We're going to talk about it as we go along on these units when we look at specialist tissue types. There's no cilia actually on this cell, but what they are is they're little sticky out bits of the membrane. They end up looking a bit like this. Like they say hair-like, so they look like hairs, but they can actually, they're more finger-like is a better description because hairs sort of implies they're dotted on the surface. They're not, they're part of the surface. They're a bit more like your fingers on your hand. Then they sort of wiggle around like this to help move stuff over the surface of the cell. And microvilli are a really good example of that. And we'll have a look at those later on as well. We find them in our intestine. They help, again, increase the surface area. You can imagine if I had stuff attached to my fist, it's not a very big surface area, but if I then had stuff attached to my fingers, there's a much bigger surface area for attachment. And that's what happens inside your gut as well with the microvilli. Okay. I'm going to stop and have a little pause because it's a lot of listening. And I want you to have a look at these questions. And if you want to, you can write in the chat if you want to give me a number and a letter if you're feeling brave. Don't worry if not. Not sure if I can, let me get my chat up so I can actually see it. If not, or if you're watching the recording, just pause the recording and have a go. And otherwise, if you're not feeling brave enough to write in the chat at home, that's fine. Write the answers that you think is correct on a piece of paper though, because it does help you to learn if you do have a go at the questions. Okay, I'll give you a minute. I'll shut up for a second so you can have a chance. So which organelle is responsible for producing the ribosomes? What controls what enters and exits the cell? And what is the main function of the mitochondria or mitochondrion? If you don't know, it's always worth a guess because you might be able to narrow it down to 2 or 3 rather than 4. And you've got better odds then if you're having a guess. Okay, I think that's everyone who's having a guess done. Let's have a look what the answers are. So the organelle which is responsible for producing ribosomes is the nucleolus. And which structure controls what enters and exits the cell? That's the cell membrane. And what's the main function of the mitochondria or the mitochondrion or a mitochondrion? It is the site of respiration. It's where we get our energy from, the glucose that we eat. So you might notice on this question, it says, which organelle is responsible? So some of these things that we've had a look at, we would refer to them as structures, cellular structures, and that's probably the safest term to use, to be honest. And some of them we refer to as organelles, which is a little bit different. So organelles, we usually refer to things that are bound in a membrane as an organelle, rather than a cellular structure. So for example, the ribosome, we wouldn't call an organelle. That's just a structure that we find within the cell. That's not enclosed within a membrane. The nucleus, though, that's membrane bound. The mitochondria has got a membrane. The endoplasmic reticulum essentially is a membrane. So we would call these things organelles that have got a membrane rather than a cellular structure. Okay, has anyone got any questions before we go on? And pop it in the chat if you've got a question. I'd just like to give you a little chance to ask anything. Feel free to put any questions as we're going along in the chat, though, and I can always pause and answer them if something comes up while we're going through. Okay. Right. So the next structure now that we're going to have a look at is a microtubule. So this isn't enclosed in a membrane. So we wouldn't talk about this being an organelle. We'd talk about this being a structure. Now, this is something that we find a lot in muscle cells. They're really important in muscle cells, and we will have a look at muscle structure later on in one of the units. So we'll learn lots about microtubules there. So these are hollow tubes. They're made of protein, and what they do is they help the cell maintain its shape. They also are quite important for things moving around the cell. So they help transport the vesicles or vesicles and other organelles and other things that move. If the cell itself moves, the microtubules help the cell to move. And when we look at things like white blood cells, we'll have a look at cells moving as well. So they're just important for structure, really. They're a bit like a, if you imagine what our skeleton is, they're a bit like that. They're a bit like our skeleton, but a bit more flexible, a bit more movable. So maybe a bit more like what our muscles are in our bodies. Then we've got things called microfilaments. These, again, they're important in the cell structure and moving things around within the cell. Again, we'll have a look at muscle cells. These play a big role. This actin, one that you can see there, plays a big role in muscle contraction. But these are very, very important in cell division. Key feature of cell division is the microfilaments. So we'll have a look at that later in the course as well. Just going to have to move my little video screen there. Okay, whoops, gone a bit too far. Let's go back. Right, lysosomes. What these are is membrane-bound organelles. So that means they're enclosed within a membrane, and they contain really powerful enzymes. So enzymes are special proteins which break down waste materials. Sometimes enzymes make things, but these ones break things. And you've probably heard of enzymes in school when you talked about digestion. You might remember back to learning about digestive enzymes like salivary amylase or proteases in the stomach, for example. So within the cell, we tend to find lysozymes, which go around looking for anything that's worn out, broken, dead, needs to be recycled and reused. So it will go around looking for things called macromolecules. That's the stuff that we're made of: fats, proteins, carbohydrates. It'll go around looking for lipids, carbohydrates, and proteins that need to be broken down and recycled. They've also got a really important role in something called programmed cell death, by releasing enzymes if the cell is damaged or needs to die. So sometimes we kill off cells on purpose, like for example, in between our fingers when we are in utero. Our fingers are joined up initially when we're developing, and we need to get rid of the tissue between them so that we can actually move off our fingers independently. So there's something called programmed cell death, like a planned cell death, where we get rid of cells. The cells that are sticking our fingers together, we need to get rid of them so we can move our fingers independently. So that's a programmed cell death. So sometimes we want to get rid of cells, and they have a role in that as well. These lysosomes, these bags of enzymes. So they're the membrane ball, really, that's found in the cell. Now, this structure now, the Golgi, it's called the Golgi apparatus. Some people call it the Golgi network, and some people call it the Golgi body, and some people just call it the Golgi. Golgi is the key bit that everyone will recognize and know what you're talking about. It's capital G because it's named after someone, person who discovered it, I guess. And you can see it's sat here next to the endoplasmic reticulum. Now, this is a better sort of image showing what it looks like. And if you think back to that lava lamp video we had a look at with the bits that easily broke off and re-merged and joined, that's exactly what the Golgi is. It's very much like this lava lamp. So remember when we talked about the nucleus, we said that it houses the DNA. It's where we find the DNA, the instructions to build the protein. And then these are the construction sites, the ribosomes, just outside on this membrane, where we build the proteins. And then right next to the endoplasmic reticulum, we find this structure here, the Golgi apparatus. And this is where we package the proteins. So instructions for building, site of building, and this is like a sorting office. We modify them. So the proteins might need a little bit of finishing off. They might need something stuck to them. They might not be quite ready for use or to be sent around the body. So we just do a little bit of packaging, get them ready for transport, and we package them into secretory vesicles or vesicles, ready to move them outside the cell or around the cell. So have a look at this image over here of the Golgi. And you can see here, we've got these bits that will merge onto the stacks, these membrane stacks. And eventually we have bits that pinch off and can go around the cell or towards the outside of the cell. So we've got a whole processing center here in the Golgi where the proteins that have just been made on the ribosomes, either free or endoplasmic reticulum ribosomes, the proteins that have just been made using the instructions from the DNA can be modified, just a bit of finishing off of the product, and packaged, ready to be pinched off in a vesicle and sent outside the cell, where they can enter the blood and transport all around the body. So that membrane, the fact that it's really fluid and can merge and break off with the cell membrane is really important. So that's what these secretory vesicles or vesicles look like. I mean, I know that earlier I said, oh, look, that's a lysosome. Truth is, you can't really tell when you're looking at a diagram like this what it is, but you will get bits like these blobs here breaking off the Golgi and merging with this outer membrane to transport substances outside the cell. A good example of that is neurotransmitters, which you've probably heard of as being important in the nervous system. And we'll have a look at them later in the course as well. So this is what happens at the cell membrane when we want to take something in. one way of getting stuff into the cell. So the cell membrane forms something called an invagination, and whatever this substance is that we need can be pinched off in this vesicle and taken into the cell. And that's called endocytosis. Cyto means cell, and endo means to bring it inside. And the exocytosis, exo, exit, to go out of the cell. This is very similar to what happens in the Golgi. So we get a vesicle containing something we need to get out the cell. Maybe it's a protein that we need to transport around the body that moves towards the cell membrane and merges seamlessly with the cell membrane, like we saw with the lava lamp, and expels the contents of that vesicle package to the bloodstream or wherever it's got to go. And that's what we had to look at ages ago when we were looking at the HIV virus of the cell. It was that sort of thing happening. Very similar. Okay, let's have a little pause and have a look at some multiple choice. Which organelle modifies, packages, and transports the proteins? Which structure do we associate with lipid synthesis and detoxification? And which one breaks down cellular waste? And if you're watching online, obviously you can just pause the video and have a go as well, if you can't watch live. You can write in the chat, but if you're not sure, just have a go on paper. That's absolutely fine as well. But do have a go because it helps you learn. And if you do it rather than just sit passively, it's more important to actively have a go. Good work. Well done, guys. Just give you another minute because there's probably some people writing in the text. Okay, good work, guys. Well done. Let's have a look at the answers. So, seeing at least one person's got 100%, so well done, good work. Okay, which organelle modifies, packages, and transports proteins? It is the Golgi body. So the rough ER, remember, it's where the ribosomes are attached. They're involved in protein synthesis, lysosomes breaking stuff down, and the smooth ER is, well, as we can see on the next question, lipid synthesis and detoxification. And which of the following breaks down cellular waste? It is the lysosome. So good work there, guys. Well done. I know this, it feels quite intense, these sessions. It's a lot of information. So well done, hanging in there. Okay, I've got another little activity for you. Ooh, let me go back. That's not what I wanted to do, give you the answers. I'm going to give you a minute to have a little bit of a break from listening to me. Just scribble on your rough paper, if you've got some with you, hopefully you have. Just scribble the numbers down and see if you can, what you can remember, what they do, their function. Don't worry if you can't remember any of it, because it's not an exam. But hopefully, I think, what have I got? They've got 13 there, and I've given you 2, so that's 11. I reckon if you've got 5, that is a really good result. First time going over. If you can remember 50%. You're doing well. So I'm going to give you a little minute to have a go at that. And then I'll pop the answers up for you. Just scribble it down. Don't worry about detailed explanations, just a bit of a scribble on what you think it does. Use the image as well to help you. That might help jog the memory a little bit. Oops. Can't see the time on my screen, so I'm trying to keep my time in using the clock on my phone. Not very successfully. I just dropped it. Oops, gone forward. Didn't mean to do that. Okay, I'll give you one more minute and then we'll have a look what you've managed to remember. Few more seconds. I'm going to pop the answers up. Right, let's have a look. So, number one there is inside the nucleus. Number two is the nucleus. So inside the nucleus, we have the nucleolus, and that's where the ribosomes are made. And number two is what houses the DNA. Number three are the ribosomes, those tiny little dots on the rough endoplasmic reticulum, and they are the site of protein synthesis. Number four is a secretory vesicle, or sometimes granule. In the notes on the course, it calls them secretory granules. This is for transport, and that includes the endo and exocytosis, the pinching off the membrane that we had a look at the process earlier. Number five is the rough endoplasmic reticulum. 6 is the Golgi body. And don't forget how close those structures are. So the nucleus, which houses the DNA, the ribosomes on the rough endoplasmic reticulum, which make the proteins, and the Golgi, number 6, which packages the proteins. They're all really close together. Number 7 is a microfilament. So that helps with this cell shape. If it's got to move, it changes its shape, things like that, and transport of substances around the cell. 8 is the smooth ER, which is lipid synthesis detoxification. And number 9 is the mitochondrium, which we're going to have a look at much later on. It's the site of respiration. Number 10, we haven't really spoke about much, is a vacuole, which is just something that's filled with water. They aren't permanent in animal cells or human cells, so we tend not to talk about them too much. You probably remember them from GCSEs when you were looking at plants, because they're really important in plants. In animals, they're not so not so important. They're not talked about so much. Number 11 is the cytoplasm of the cell. So that's the sort of gel-like structure. Oh, it wasn't 11. There was 14 altogether. Number 12 is the lysosome. So it's impossible to tell which is which between 10, 11, and 12 because they all just look the same. But the lysosome, so it doesn't really matter which way round you've got them, if you've got any of them. The lysosome is something that contains the enzymes to break down the cellular waste. Number 13 is a microtubule, which is important for maintaining the cell's shape, and it's got a role in cell division as well. Transport as well. It's like a sort of a skeleton slash muscular system equivalent inside the cell, if you like. Movement, shape, transport. And 14, probably the most important of all, is the outside of the cell membrane. Right, who's feeling brave enough to count up and tell me how many you got correct or almost correct? It doesn't have to have the exact meaning down that I've included, but something along those lines. Give me a score if you're feeling brave enough. Tell me how many you got out of 14. I reckon if you've got 5, you're doing quite well there for a first look. Anyone got 5? $11. Wow, that is impressive. Excellent. Well done. That is really good. How about anyone else? Anyone else feeling brave enough to tell me? Fair enough. If not, I don't know how many I'd have got on a first go through that, to be honest. It's not easy. There's a lot of information. So really, really good getting 11. That's fantastic. Okay, let's have a go at multiple choice. It's a little bit easier. What's the mitochondria or mitochondrion, if it's one? Which statement is correct? It's about the endoplasmic reticulum. Which of the following is not bound by a membrane? And let me move my little bar again. What is the function of the chromosomes? So have a little go at this, and I'm going to have a look at some. I'll give you a couple of minutes to just quickly read through that. I'll have a look at some equations in a second. You can pop your answers into the chat if you want, but you don't have to. Lana, you don't remember, to be honest, with the structures. That's fine. I honestly don't think I'd have remembered much when I first went over it either. It's very hard. And it is just a memory test, essentially, at this point. And it's important that you understand it, really, not have an amazing memory. Okay, I'm going to pop your answers up then. I can see 1 or 2 answers going in the chat, but not too many. Maybe getting a bit tired. It's a lot. We're getting towards the end, guys. You're hanging in there. So what does the mitochondria do? It's involved in cellular respiration. That's breaking down our glucose to get energy. Which statement is correct? The endoplasmic reticulum can be rough or smooth. Which of the following is not bound by a membrane? It's the ribosomes. They've just got a small subunit and a large subunit. And what's the function of chromosomes? They contain short sections of DNA, which are genes, and that encode proteins. Okay. If you've got any questions, now might be a good time to pop them up for me, and I will You can type while I'm talking, and I'm just going to have a look at the next section, which is calculate the sizes of the subcellular structures using appropriate units. So you need to know your units. So we've got 10 to the minus 3 meters, or 0.001 meters is a millimeter. Then 1,000th of a millimeter is called a micrometer. And then 1,000th of a nanometer is called, of a micrometer is called a nanometer. So I want to show you this equation triangle, and I'm not sure if you've remember equation triangles from school, but with an equation triangle, what you do is this number here is divided by this number here, or this number here, and these two are multiplied together. So what do I mean by that? Well, if you're trying to find something, you cover up the thing you're trying to find. So if I was trying to find the magnification, M stands for magnification in this equation triangle, I'd cover, if I was trying to find the magnification, I cover up M, and then I divide I by A. So I stands for image size and A stands for actual size. So let's have a look. A student looks at an image of a plant cell under a microscope. The image measures 45 millimeters in length. So that's the picture she's looking at. The actual length is 0.015 millimeters long. Calculate the magnification. So I'm trying to find the magnification. So I cover that, and that leaves me with image size divided by actual size. So magnification, the thing I'm finding, I cover that up with my hand, and that leaves me with image size over actual size. So that's the equation I'm going to write out. Magnification is image size over actual size. The image size was 45 millimeters, and the actual size was 0.015 millimeters. So just type that into a calculator or do it in my head. And the magnification is times 3,000. So the important thing with this calculation is to get both of those in the same unit. They were given to me both in millimeters in the question, so that's good. And magnification doesn't have a unit. It's just how many times something has been multiplied. Okay, next question. We have a photograph of a bacterial cell is shown in a textbook. The magnification is times 10,000, and the image measures 25 millimeters. Calculate the actual length in micrometers. So I'm trying to calculate the actual size. So I'm going to cover up this part of the equation triangle, and that leaves me with image size over magnification. So I'm going to write that down. Actual size is the image size over magnification, and I'm going to do 25 millimeters over 10,000 because that's given in the question. The image measures 25 millimeters. The magnification was 10,000, and that's going to give me 0.0025 millimeters. And you can convert it to micrometers if you want. And its actual size is 2.5 micrometers. And another example, we've got a drawing of a human red blood cell with an actual diameter of 7 micrometers. The diagram has been drawn at a magnification of times 2000. Calculate the image diameter in millimeters. Okay, so I'm in micrometers here and I need my answer in millimeters. So I'm going to have to do a bit of a calculation to get my units the same. So the image size is what I'm trying to find. So cover that up, and that leaves me with actual size times the magnification. So if we do the magnification, which was 2000, times by the actual size, 7 micrometers, the image size is 14,000 micrometers. And 14,000 micrometers is equal to 14 millimeters. So the image diameter is 14 millimeters. So hopefully that will have covered a lot of what you should have in your first assessment, which your course tutor is going to go through with you very shortly. And there you go. That's the bit there in bold that we've covered today. So have you got any questions for me just before we go? If you have, you can pop them in your chat. If not, I will see you on Thursday. You're all quiet? No questions? Okay. Thanks then, guys. I'll see you on Thursday if there's no questions.